Right. So in the earlier lecture this morning, I tried to give you an overview of 16S bioinformatics. What we're going to move on now is to shotgun metagenomics, where, as I said, the essential difference is this part of the analysis pipeline would all be the same. We're taking a sample, extracting DNA, but it's how we sequence that DNA. So rather than focusing on this single marker gene, we're going to generate sequence from all the different members of the community. Now, in 2017, we wrote this review paper where we reviewed all steps actually in a metagenomics analysis. And at that, we talked about two different strategies for analyzing the sequence. One we referred to as assembly-based, and the other is read-based. And in fact, when we wrote the review, I was responsible for the assembly-based because actually I was in a bit of a niche in the field. Most people didn't do assembly-based analysis at that point. They did everything read-based. And Nicholas Agata wrote the read-based proportion of this review. And in the rest of this talk, I'm going to talk about read-based methods for analysing metagenomics data. But actually, since we wrote the review, probably the field has shifted until the majority viewpoint is assembly-based. And that's what we'll be talking about in the rest of the course, is the best way to analyse metagenomics. But I still think there is a place for read-based analysis methods. And I also think, through my career, one thing I've noticed is that at various points, people want to be dogmatic about what is the best way to analyze a microbial community in terms of what is the best data, what are the best methods. But my experience is that there isn't any single best way. All the different methods have pros and cons, and you should be cautious or skeptical of people that tell you this is the only way to do it. Because really they probably have an underlying agenda, okay? Either they are, it's about sort of controlling the field. There are people that will tell you have to use my method. And I think that in reality, every method will tell you something useful. You just need to gain experience in interpreting the different methods. So for that reason, we are going to go through read-based methods, and we're going to start with taxonomic profiling. So just like you can taxonomically profile with 16S reads, you can use your shotgun metagenome reads to profile the different members of a microbial community. And essentially, it's the same principle as when with 16S sequencing. We're going to take each read and try and assign it to a taxa given a database of known taxa and sequences. The fundamental difference, though, is the fact that we haven't got a single marker gene, but we have to have, for the most simple, for most methods, we need to have the whole reference genome in our database. There is exceptions to that. There's a method called MOTUs, where you don't profile against the entirety of the genome of the different members of your communities. Instead, they've actually constructed these MOTUs through a method very similar to binning, and then and those MOTUs only use a set of 40 marker genes. And we'll talk about marker genes the rest of the course. For marker genes here, we mean single copy core genes. Okay, these are genes that are present in every prokaryote in a single copy. So in MOTUs, they take 40 of these single copy core genes. Over a variety of data sets, they look for correlations in them. They link them together into what they call MOTUs. And then rather than profiling against reference sequences, you profile against these MOTUs. Plan is also based on marker genes, but it uses a different principle, and I'll sort of go through that in a bit more detail. it tries to find marker genes that are unique to a particular set of taxa. So at different levels of taxonomic resolution, it's looking for genes that enable you to discriminate that group of taxa against the other groups. So in a way, it's the sort of opposite of MO to use, which is using these core genes that are shared by all organisms. It's trying to find unique genes for a particular of organisms. And I'll talk a little bit more about the algorithms you then use for comparing against those databases, but I won't go into too much detail because I think you did do this last week. And I suppose, though, the fundamental difference with 16S is that when we've got shotgun metagenomics, we can also now do functional profiling. So we're not just restricted to saying what taxa are present in the community. We can say what functional genes with what abundances are present in the community. But then it's pretty similar. What you actually then need is a database of functional genes to compare your sequences to. They can be generic databases which try to include every possible functional gene. So PFAM would be an example of that. But then there are also more specific databases for a particular targeted, that they're aiming to get a particular set of, gene functions associated with a particular type of metabolism. So KZ would be a good example of that, which curates carbohydrate metabolism genes. There's another thing called COGS, which turn out they're quite old. They've been around for 20 years or so, but they're useful because they were so well curated to begin with. So COGS aim to find clusters of orthologous genes, so genes all doing the same function. They're not very representative, but because the clustering was done by hand, they're quite reliable. So actually, for our single copy core genes, we use COGS to define them. And I think that's quite common. But they're trying to find a set of orthologous genes which share a common ancestor, and then they're very good for building phylogenies. But as I said, they aren't super representative. So most genes will not be assigned to a COG. There have been attempts to do, to expand on that with some automatic generation. And that would be eggnogs. For functional annotation, we often end up using a database called KEGG. That stands for the Kyoto Encyclopedia of Genes and Genomes. The nice thing about KEGG is actually the diagrams. So it has a it has a hierarchical structure, just like taxonomic classification. So you have a sequence which gets mapped to a gene, that gets a KEGG ortholog number, which essentially has a one-to-one, well, maybe not a one-to-one mapping, but can be mapped onto EC numbers, which are enzyme classification numbers. But those KOs are linked at a higher level into modules, all of enzymes, all performing a similar function, which is useful in a lot of contexts of microbial community analysis. And then those modules are linked into higher level units called pathways, which is essential. I'm not quite sure the distinction between a module and a pathway. A pathway, I suppose, is a higher level grouping. These things perhaps are not well defined, but they are very useful in terms of analysing your own data. The other nice thing about the KEGG database is it's got enzymes, but it's also got it's got compounds as well. So if you're trying to incorporate other forms of data, like metabolome data, it's quite easy to do that in the context of KEGG. Metapsych, though, is a similar database. So here's an example of a KEGG module, biotin synthesis. You've got the different genes involved and the different compounds. And then that is then embedded within a larger pathway of biotin metabolism. Okay. So I'm very briefly going to talk about how you search against these functional databases, because you did this in the context of genomics. And there isn't really a lot of difference, except for when you're thinking about metagenomes, you do have to be concerned with speed. So you can have a lot more data if you use the best possible methods, which are probably still homology search, then it will take a long time. But when we still do use homology searches in some situations, there's a very nice, efficient program called Diamond. So did you use Diamond last week, the week before?
No.
Okay. All right. Well, it is worth talking about these programs then. If you're going to do homology searches at large scale, Diamond's about the best possible method. Now, when you're searching against the databases, you do have a choice. You can do this in nucleotide space or in amino acid space. So basically, either you take the DNA sequence itself to search against your database, or you convert it to amino acids first. Now, if converting to amino acids, unless you've already done gene calling, you will have to search in all three possible translation frames. So that's what BLASTX does. And Diamond, for instance, has a version of BLASTX where you can search in those three frames, but it's much more efficient than BLAST. So yeah, because I didn't want to go into this in too much detail, I tried to do a little summary of all the different sequence search algorithms and how they sort of compare to each other. So the most basic methods, as I said, are based on sequence homology, and they are both sensitive in that you can find things that are very distant to your search query, and they have a lot of precision in that you can distinguish between two queries which are quite similar. The drawback is that they are slow. Now, a much faster method is based on just using k-mers. Okay. Now, k-mers, and I'll talk about these in a bit. I guess you've done k-mers, right? Surely.
No.
Okay. All right. Well, we'll talk about k-mers quite a lot, particularly in the context of both in terms of clustering sequences, binning in metagenomics, but also for metagenomics assembly. So I won't go into them in too much detail now, but essentially what you need to know is that a k-mer is a nucleotide word. Okay, so it has a length, that's the k, so it might be 4 or 5, and then you're defining your sequence in terms of little words with that length of nucleotides. And as I say, we'll go through this later in the course, but in terms of searching, they have the advantage they're very fast, okay? Because if you decompose your query into k-mers, and you've got a database decomposed into k-mers, you can use hashing methods to find how much in terms of k-mers your query overlaps with what's in your database. Hashing methods, where you basically say, do these two things share the same k, are very fast. The problem is they're not very accurate, okay? Because if you have an error or evolutionary divergence, it will break up the k-mers. There's a reason we want to do alignment, okay? But what you can do is sort of use the k-mers as a first step to find possible hits, and then you would do a more accurate method. So this is what vSearch or usearch does. It's also essentially, I think, what Diamond does. Now, you can get a method that has high level of precision, but is also fast, and that would correspond to mappers. And they're based on sort of quite clever algorithms like Burrow-Wheeler's transformation, or more clever uses of k-mers. So I probably will talk through this tomorrow, but you can use a thing called minimizers, which is a clever way to select k-mers, and that gives you things that are both fast but also precise, in that you can distinguish between very fine scales of difference, but what they tend not to have is sensitivity. So if you're too far away from the thing in your database, then the mapping methods won't be reliable. Another fast method that has sensitivity are hidden Markov models. And I won't explain how they work, but from your viewpoint, I think it's just worth knowing that they're very useful when you want to do a distance search, and that's often the case in functional annotation. The function of a gene doesn't depend too much on its fine scale difference. Just on a sort of, but that they have the sensitivity of homology searches, whilst being much faster, but they don't have the precision. So yeah, so that's the different algorithms, and we can talk about different bioinformatics tools for mapping. Examples would be Bowtie, BWA, or Minimap2. Hidden Markov models, the standard one is Sean Eddy's software, isn't it? Which I think is called Hammer. Yeah, so hopefully you'll have an opportunity to see some of these different. Okay, so that's the different algorithms. All the software that's out there basically consists of one choice for some of those algorithms and one choice for a database. So for example, Megan, which I'll talk a little bit more about, is based on homology search with Diamond against the entire NR or NT database. Kraken uses k-means against the user-defined, or they've also pre-compiled refseq databases. Yeah, and I haven't used it. We don't use this much because I think Kraken is generally better. But there's a program called Centrifuge, which is using its own form of mapping based on an FM index, and I guess coupled with the refseq database. So all these methods, they try to classify every read. The marker gene-based methods like Metaflan or MO to use. One thing to bear in mind is that they will give you the profile of organisms or functions in give you the profile of organisms in the community. But because they're using a subset of the genes, they won't classify every read. They're only going to classify a fraction of the reads. Okay, so how does Megan work? It's pretty simple, really. So it would use the entire NR, and then it depends upon this very efficient search program produced by Daniel Houston Diamond to do a BLASTX of your metagenome sequences against that. Then it takes, so this is exactly what I talked about in the context of 16S. It takes all hits above a certain level of identity, and then it does a lowest common ancestor algorithm on those hits. So in this instance, we've got hits to 3 different species of bifidobacterium. We'd end up classifying this read as deriving from bifidobacterium, but we'd say we weren't able to be more precise. And when you've got short reads, they will usually be a lot of imprecision. You wouldn't expect to be able to get species level classification in general from a short read. And yeah, and then we attach the rest of the taxonomy to that hit. And yeah, if we had multiple, if we had all the hits from the same species, so they were all biflongum, then we could be more precise in our classification. Kraken does something similar, but it's all based on k-means, which makes it very fast. And there's definitely, an appeal to fast things. When you're doing an analysis, oftentimes you'll run, say, Kraken to get a quick idea of your data set, and then maybe you use a slower taxonomic annotation or perform assembly, whilst, it gives you something to work with whilst you're waiting for those slower methods. But the basic principle with Kraken is that they've actually pre-computed the LCA on k-means from their reference database. So for every k-mer, and they use k-mers of length 31, so that's quite a long k-mer, so there'll be a lot of them. For every k-mer, they've worked out what species it appears in. So some will be unique to species, and they will be, I guess, at the tips of the taxon tree. Some will be shared between species, so we've got an orange one here, or shared at a higher level. Okay, and then you take your read, you just look for those k-mers, and as I said, that's very fast searching for k-mers. And then for each read, you'll get a count on the tree of how many k-mers at that level of precision were observed, and then your assignment is just basically the highest weighted path from the root of the tree, so this is the root of all things, down to the tips of the tree at the species level. And yeah, it's a pretty robust method, to be honest. There is now a Kraken 2, which I think focuses more on unique k-mers. It's relatively easy to create your own databases. You just feed in a set of genomes, and it's got software for generating the database files from that. They tend to be quite big. It is quite a bit of memory, but on the whole, it's a super useful program, as is Metaflan actually. So Metaflan is as I mentioned before, is based on identifying genes that are unique to a particular taxonomic group, and then having done that, you basically just use a mapping algorithm which is fast. I think they use Bowtie actually, to map onto the set of genes that they've pre-calculated as being discriminatory to particular groups. Yeah, and this is quite old. The database now, I'm sure, is much bigger. In our experience, Metaflan works really nicely for human microbiome data, probably because we've got a lot of genomes. Yeah, it tends to be a bit less useful well, in fact, not useful at all sometimes, where you have you have environments which are less well catalogued in terms of genomes. But for human microbiome work, Metaflan is very helpful. So see, this is actually taking a figure from a paper that both Mark and myself were involved in. But one thing you always need to bear in mind when doing taxonomic profiling from metagenome reads is false positives. Okay. In actual fact, and this is one of those examples of why metagenomics can be trickier than 16S. I think the issue with false positives is even greater in metagenome data. And I think it comes down to the fact that different parts of a genome evolve at different rates. So there isn't sort of one cutoff for any part of the genome that gives you, say, species level or strain level. You really need to consider the whole genome. Okay. So a classic example of this is this New York subway paper from 2015, where they claimed to find plague. And that was based on the output of Metaflan, in fact. But before Metaflan did a database update where they removed prophages. So what they really found on the New York subway was a prophage that was found in the Yersinia pestis genome. And if you think about it, the odds of a prophage moving between species is very high. And that is exactly what had happened in this case. They hadn't really found Yersinia pestis. They'd found another species of Yersinia that had picked up a prophage from a genome that was Yersinia pestis. And I mean, yeah, at least I really can't, I shouldn't talk about this too much, but the authors of this were probably aware of that fact before they went ahead with the publication. But, you know, with that sort of claim, you get it into Cell. If you've just found regular Yersinia, you don't get it into Cell. That's one of the sad things.
You get a retraction, though, later on. Surely you get a retraction.
Did they end up retracting that?
I don't know. They should have done, shouldn't they?
Yeah, they should have done. Yeah. You know, I was told they actually spoke with Nicholas Agata, who told them that it's not Yersinia pestis. But anyway, let's move on. So a good way to deal with these false positives is what we did in this 2013 paper. So these ideas have been around a very long time. You take a genome, ideally close to the organism that's in your samples, and you just look at the coverage across the genome. So here we've mapped onto, this was from an outbreak of E. coli with a Shigella toxin. This was that German outbreak, you know, which was later traced to bean sprouts, wasn't it? I think.
Yeah.
Certainly some sort of sprouting.
I had a talk on that.
But anyway. Okay, so that's nice. So Mark will talk about this a little bit. But the point is, here, you know, we've mapped all the reads. We've mapped all the reads from all our samples onto the genome that we think might be there, and we can see that we've got coverage across the whole genome. What's really nice is that you can see you've got higher coverage on the Shiga toxin, on a prophage containing the Shiga toxin, which illustrates that sometimes that phage is going lytic. So it's really fascinating. You can also see something else from this diagram. So why do you think we've got higher coverage here and here to the middle. Does anybody have an idea?
They have been told that.
Well, let's hear it then. Somebody should know.
Come on, someone shout out the answer. Closer to the origin, someone's saying.
Okay, all right, yes, that's right. And I'm not going to talk about it, which in detail, so I want to mention it at this point. There are actually some methods that utilize this. So this is called the peak to trough ratio, and the magnitude of that ratio will reflect how rapidly the organism is dividing. So it gives you a really nice way, or potentially really nice way, of estimating growth rates independent of any other method. And so there's quite a lot of metagenomic software that takes these peak to trough ratios and estimates the growth rates. But we're not going to be doing it in any of the tutorials, so I just wanted to mention it as a thing you can do. Okay, right. So I talked a bit about taxonomic profiling and the software available. Functional profiling, exactly the same principle, but as I mentioned, now you need to be able to do searches that are more distant, perhaps. So examples of that software. So Megan gives you both taxonomic and functional profiles, and it can do that because on the NR database, the non-redundant protein sequences, a lot of things are annotated to function as well as being annotated to taxonomy, and it's integrating all that information. And actually it does then map them onto KEGG orthologs, so you can get all the same, you can get KEGG profiles out of that as well. Another choice for doing functional profiling is human. In actual fact, we might be Luddites, but we don't tend to use any of these pipelines. We just map onto a functional database and integrate the information ourselves. And I don't sort of think there's any obviously better way for doing things than that. All right, so I've just now got a few slides showing you some results from breed-based profiling. And this is actually the same set. It's the same data set as I showed for the 16S analysis. This is the CICRA study where we've got individuals being treated for Crohn's disease, but now I'm looking at the metagenome data. So we had 137 samples in total. The median number of reads was it's about 6 million, and we did 2 by 150 base pair sequencing. So I subsampled to 1 million reads, and I did this as I explained on Friday. If you're ever doing diversity analyses, it's a good idea to use similar read numbers across samples. And here I wanted to compare to the 16S estimates of diversity. So I used Metaplan to assign species, but to get species profiles. And here I'm showing the species Shannon entropy. And on Friday, I'll also explain what I mean by the Shannon entropy, but for your purposes, it's just a method, just gives you a measure of the diversity in the community. And we see the same thing as we saw for 16S. Basically, the effect of the treatment is to depress the overall diversity. We then went on and also looked at functional diversity, mapping against the KEGG. And that's sort of interesting because we don't see the same reduction in average functional diversity. And just to emphasize also, healthy versus time point A at the start of the diet, we see a higher species diversity in healthy. What's quite interesting is when you look at the functional diversity, it's actually higher in the Crohn's individuals. So it probably says something about dysbiotic microbiome, because these things are perhaps doing a wider range of pathogenic functions. But yeah, anyway, it's quite interesting, and I think illustrates what you can get out of functional versus taxonomic profiling. Okay, so summarize then. This is what we were debating with Mark. I still think 16S has a role in microbial community analysis. One thing I didn't really get onto is there's no reason why you can't use other marker genes than the 16S. Now, the reason we don't is because we have these huge 16S databases. But as we get more and more MAGs, we're effectively creating big databases of other marker genes. So there is an argument that there might be a role for some of these alternative markers. And one we published a paper on was RPOB, right? And they do have advantages over the 16S in that you don't have the issues with confounding copy numbers. So you choose a marker gene that is single copy. So all this profiling is strictly reference-based from metagenomics data. OTUs is a non-reference based method for 16S. What we'll talk about next is non-reference based methods for metagenomics based on you could actually go, I didn't really talk about it, you could go down below the species level to strains. I think I will talk about this tomorrow, but that depends upon having those strains in your reference data set. Okay, so how are we doing for time? Maybe I can go straight on to my next talk, Mark. Is that?
Yeah, I think
try and get it.
Probably that's the most efficient way to do things.
Try and get it done in the hour.
Yeah, okay. I think so.
Right. So got to find it. Binning principles. That's it. Okay.
I mean, it's worth just people got clear in their head the difference between reference-based profiling methods and binning methods. So with the reference-based profiling methods, you have to have reference sequences, you have to have genomes out there, and you say, all right, I know what E. coli genome looks like, I know what Salmonella enterica genome looks like, and by comparing what I've got against, say, those two genomes, I can work out this is more likely to be E. coli than Salmonella. You have to have that prior information. Now, obviously, if you're just comparing between two, it's a very simple thing, but if you're comparing against all the genomes and all the species that are in the databases, there are tens of thousands, hundreds of thousands of those, it becomes more complicated in practice. But in principle, it's simply that of like, I've got all these references here, which one am I calling as being present in this read or in this set of reads and so forth. Whereas what we're talking about now is where you don't necessarily have to have any reference at all. There's this Latin phrase that people use called de novo, where you're just doing the thing without any prior information at all. So 16S is in effect de novo in its practice because you can find new sequences in there that you've never seen before. It does rely on one assumption, which is that the sequence barcodes that you use to make your primers are actually conserved, and that requires the prior knowledge that people have had over years of aligning like the 16S so that you can see what are the conserved regions and use them. And there are debates about whether those so-called universal primers really are universal. So 16S kind of combines both approaches in a sense. But
well, 16S is de novo based if you build OTUs. Okay?
Yeah, but you have to know what 16S looks like, though. You have to know what the conserved sequences are that you're building primers from.
I suppose I use a different feeling of de novo versus.
Well, no,
de novo is where you can find a new taxa. References where you can only find taxa that you've seen previously.
Yeah, you're right. I'm just spreading it out.
And there was a lot of debate about this in 16S analysis because reference-based does have the advantages. It's much less sensitive to noise, okay? But whenever you do a study, the de novo-based methods would give you more statistical power, okay? And I think we're going to end up with the same debate in metagenomics soon because we are getting really big MAG databases. And the question is, perhaps you just compare it to those databases. But I still believe that de novo-based approaches for individual studies will be the most powerful.
Well, the question is whether you're looking at something that's already mostly known and you're using metagenomics or these approaches as a quick way of just getting a census, or whether you're looking at something where there's a considerable fraction of it that's never been seen before. So it depends on if you're looking at the human gut microbiome, that's mostly a known thing now.
Okay, but that also depends on what level of resolution we're talking
about. Exactly.
I believe that every human gut microbiome contains a unique strain of a large proportion of its species.
It's all
but
about, it's like looking through a telescope and adjusting the focus to the near or to the far,
I think the telescope is optimistic. There's a phrase from the Bible, isn't it? To look through a glass darkly. I think that's a better way to think about it.
Yeah.
Because that in those days mirrors were terrible. So
Yeah.
all right. So look. So metagenomics assembly and binning. So this is going to be a bit more complex. All right. So binning, is something that should be on everybody's radar now. It really has transformed microbial ecology, essentially because it enables us to get genomes from uncultured organisms. So this is quite an old tree now, from 2016, but this is in this tree they've used a combination of MAGs and reference genomes to attempt to reconstruct tree of both prokaryotes and eukaryotes. And in terms of 2016, every red dot here corresponds to an entire phyla for which there were no cultured representatives at that point. So there's huge amounts of diversity that have been revealed by metagenome binning, and I'm going to talk to you about methods for automatic binning from metagenomes. And again, as I mentioned at the start of the other talk, this has been labelled as one of the milestones in human microbiota research in the context of generating large-scale binning collections from the human microbiome. And I might talk about one of those tomorrow. Okay. But before we talk about binning, I'm going to talk a bit about metagenomics assembly, because that would be the first step before binning, and it sort of explains why we need binning. And then I'll talk about how we do how we do binning, and then I'll have a brief set of results from that same Crohn's disease data set that we looked at. So now we're on this part of the bioinformatics pipeline. We're going to do assembly and binning of contigs into MAGs, metagenome assembled genomes. Okay, so what is assembly? So when we've got short reads, I suppose it's pretty straightforward, really.
They've done assembly on culture genomes already.
Okay, so did you talk about different algorithms for assembly? Do you know what a De Bruijn graph assembler is?
No, we didn't go into the details of the algorithms. We
Okay,
just
well,
used the pipelines.
I mean, did you even talk about? So when would
we didn't talk about
say
graphs? No.
Pardon?
We didn't talk about graphs. So yeah.
No. Okay. All right. I'm going to ask them a question though.
No, we did actually. You did show.
Say you're
you did see
assembling
some graphs. Yeah, a little bit.
If you're assembling a genome, say you've sequenced the genome, and you get contigs, a fragmented assembly. For what reasons could your genome end up fragmented?
Who's still
Any
awake?
ideas?
Who's still awake?
Why would you not? Pardon?
I'm just saying, who's still awake? Who wants to answer?
Why wouldn't you get the complete genome?
Say it loudly enough for him to hear.
Repeat regions that can't be resolved.
Repeat regions that can't be resolved.
Okay,
Short reads.
So how big would a repeat region have to be to fragment your genome?
How big? 300 base pairs.
Well, yeah, that's the correct answer for a short read assembly. Okay, it basically needs to be longer than your read length.
Yeah.
Okay, and that's the sort of definition of a repeat. It is sort of defined by your sequencing technology. Is there any other reason why your genome may end up fragmented? There is only one other reason, really. It's sort of obvious.
I think there are two of them. Coverage?
Well, Seb, you can't answer this. Really? I mean,
What?
Any other reason why your genome would be fragmented other than repeats?
Gaps in the sequencing.
And what causes gaps in the sequencing?
Well, there's two things. He's already asked one of them.
Okay. Well, yeah. So repeats aren't gaps in the sequencing.
No.
Repeats
But
are inability.
Seb
to
answered the
resolve.
question by saying so this the coverage. So it's just statistically, if you go in with low coverage, there will be parts of the genome you just miss just by bad luck. So you end up with
Yes, that is true, but it's also worth bearing in mind that different portions of the genome will have different biases for coverage.
That's right.
So, in particular,
Chemistry.
high GC will result in low coverage, more chance of fragmentation, but also sometimes the actual, depending on how well you've done your sample preparation, I believe the actual physical structure of the chromosome can prevent you getting reads sometimes. So even with genomics, we end up with fragmented assemblies for those reasons. Okay, all right, so go back to the theory of assembly. Essentially, it's quite straightforward. We're looking for overlaps between our reads that enable us to join them together into longer sequences. So this, would be a very simple example. We've got two reads, they overlap, we can make a longer sequence, easy. Now, we're going to go into this in more detail when we do long read metagenomics, but in principle, there's two sort of major different types of assemblers. One are known as overlap layout consensus, and the other De Bruijn graph assemblers. So in overlap layout consensus, you basically it's sort of how you would assemble something if you were a human being, in a sense. Well, sorry, you are a human being, but it's sort of the most intuitive way of doing things. You look for overlaps between your reads, and that forms a graph, and then you just try to find longer paths through that graph, visiting each of those overlapping regions. The challenge is that, there's two challenges with this. Firstly, you need to compare every read to every other read, and that can be quite slow. I mean, we can use the same principles that we developed for mapping algorithms to speed that up, things like minimizers, but it's still slow to compare every read to every other read when you've got short read data and hundreds of millions or billions of reads potentially. And then actually processing the graph is difficult. That's where the consensus part comes in. You typically consider it locally, try to find a consensus that accounts for errors, because errors will create a lot of structure in this graph. It's difficult. Now, it's actually come back into fashion again because it scales really badly with read number, but when we've got long reads, we don't have a lot of reads. So OLC assemblers, although not typically used for short reads, we'll return to them again for long reads. For short reads, almost certainly the only type of assembler you need is something called a De Bruijn graph assembler. It's a fancy name. I won't go through the details. It's nicely explained by this blog post here, actually. So if you want more of the details, I'd recommend you go and read that. But it's basically assembly at the k-mer level. And because it's purely based on k-mers, just like k-mer matching for sequence searching, it's really fast, okay? So we decompose each read into its constituent k-mers. So these k-mers are these little nucleotide words. So, here we've got trimers, okay? So our first read contains the k-mer ATG. It also contains the k-mer TGC and GCT and CTA, okay? So we form our graph, well, actually out of the K-1mers, so the TGs, the GCs, etc. And then we link them if we have a k-mer that is present in a read, okay? You form a graph like that, and then the sort of magical thing is that taking paths through this graph will assemble your reads. And you've done it without having to do any form of slow, pairwise, sequence-to-sequence comparison. So it's super efficient. but it does have drawbacks. So if you think effectively, the drawbacks are that it can only deal with exact overlaps. So it's not so good for noisy sequences, all right? But it's ideal for generally highly accurate short reads. There's also an issue with the Kamer length, okay? So for different levels of coverage, there's a different optimal Kamer length. And if I had a whiteboard, I'd explain this to you. But you can sort of convince yourself that if you have high coverage, then the average overlap will start to approach the length of the read themselves. Okay, so if imagine piling up lots of reads on a genome, then you're going to get long overlaps. So for high coverage genomes, you want high Kamer lengths because they give you the best chance of resolving repeats. But for low coverage genomes, you actually want smaller Kamers, because when you have low coverage, the overlaps will get smaller, all right? And you'll miss overlaps if you go straight to a high Kamer length. So the big sort of breakthrough in de Bruijn graph assemblers was the use of iterative Kamers. So rather than just using one Kamer size, you start with a small Kamer size and you go to increasingly larger Kamer sizes, feeding back the results from the earlier assemblies. And the ones you'll do in the tutorial are all based on iterative Kamers. Okay. Now, essentially though, short read assembly is a simple process. You construct this graph, and it's pretty similar whether you've constructed the graph from de Bruijn graph principles or from OLC. And then you basically just process that graph. So if you see tips on the graph, it's likely an error, okay? Tip is likely an error because a tip will form when you see a base change in one read where you don't see it in any other read, okay? A bubble will probably correspond to variation. So you did variant detection last week, didn't you? With programs like VARScan, okay? If you've got a metagenome with multiple strains in, what you'll see is bubbles in your assembly graph, with one path corresponding to one variant and the other path corresponding to the other variant. So basically, all assemblers do is they'll trim off structures like that, the leaves, then they'll make some sort of decision on the bubbles. If a bubble is long enough, and it might also look at the abundance of the two possible paths. If one path is of low abundance, then it might break the bubble or pop the bubble is how they refer to it. So you process the graph in this way, and then you output contigs, which are basically just linear portions of the graph. Okay, so where you've got regions where each node has one edge incoming and one edge outgoing, then you're confident about the assembly at that point. You'll never see it in short reads, but you will see it in long reads. What we really want is entirely circularized genomes, but you won't get that in metagenomics. And the reason is, as we talked about, it's basically repeats. So the uncertainty in an assembly is when you have repeat regions which are identical at multiple places in the genome. But then what makes metagenomics assembly difficult is that you'll also get repeats between genomes, and that corresponds because you might have multiple strains of the same species. So whenever the repeat exceeds the read length, then the path through the graph becomes uncertain. And I'm illustrating that here. We've got 3 reads, that's the red arrows. So we've got a read running into this first repeat region and then running out of it, and then we have another read running out of the second repeat region. When we come to assemble, we don't know whether we should go through there, that's correct, or actually through there, into there, and out of there. So the assembly graph is a way to represent that uncertainty. So here we can see, so typically and I'm going to go into talk about assembly graphs because we'll use them again tomorrow in strain resolution. Each node on the graph corresponds to a sequence. The edges are overlaps between those sequences, and then actual true sequences are paths through that graph. So here, as I say, we've got this bubble structure representing variation. We've got two possible paths through here. If we go through this path, we introduce a G, and if we go through that path, we introduce a T. So all the assemblers will produce these assembly graphs, and then, as I said, they just process them and output contigs. Okay, so I mentioned this previously. The reason why metagenomics assembly is so difficult is because of strain assembly. So here actually I've taken, so this isn't real data, it's synthetic data. So this is the assembly graph produced when you try to assemble 6 strains of Lactococcus lactis at the same time. So you can actually see each colour in this graph corresponds to a different reference sequence. And if you looked at it really carefully, you could probably convince yourself there are 6 paths through this graph. But because of the repeats shared between the different strains, it becomes incredibly complex. So assemblers, they would just output those linear regions as contigs. So what can you do about that in terms of your analysis? And that's where mags come in. So mags, essentially, we're looking for features that are shared across contigs that derive from the same species, that then enable us to cluster the contigs back together into the genomes. Okay, so one possible feature that you can use is sequence composition. So this isn't quite the same as when we're doing taxonomic classification, because effectively that's too fine scale a resolution. We need a feature that is more general to the genome. So typically what we'll do is k-mers again, but relatively short k-mers. So this is just, this is exactly the same as the De Bruijn graphs. If we have a sequence here, and we want to decompose it into bimers, so k-mers of length 2, we just basically pass through the sequence and count up how many occurrences of a particular k-mer we observe. So in this sequence, we see AC twice, so that gets 2. We see CA once, etc. That's our k-mer decomposition. There is actually a subtlety to this, though, in that when you do any form of sequencing, you'll get reads from both the forward and reverse strand. Okay. For both 5 to 3 and 3 to 5 direction, we don't want that to confuse our analysis. So all you do actually is you basically use something called canonical k-mers. So you treat the k-mer and its reverse complement as identical. So in this case, AC would be combined with TG, basically. Now, for binning contigs, tetramers are about the most useful k-mer size. So that's a k-mer of length 4. So that would give us 256 unique k-mers. But because, as I said, we use canonical k-mers, we actually end up with 133. It's not 128, i.e. half 256, because of palindromic k-mers. So a palindromic k-mer is basically one that looks the same both in terms of its forward and reverse complements. But you can read about all this stuff if the technical details are of interest. Okay, so here I'm showing how clustering by sequence composition works. I've taken 5 different species and I've broken them into 10 kilobase pair pieces, so 10,000 base pairs. I've decomposed them into tetramers, and then actually I added on pseudo counts and log transformed those k-mer profiles, and that enabled me to do principal components analysis, which again we'll talk about on Friday, as a means to plot those sequence fragments into 2 dimensions. Okay, and what's nice is that we can see distinct clusters. So this lactobacillus nicely clusters away from the other lactobacillus species, There is, though, quite a bit of overlap. So the sequence composition by itself doesn't have enough information generally to cluster out at the species level. By the way, it's an interesting thing to think about is why is this the case? Why do sequences from the same genome share these similar patterns of k-mer frequencies, even though you've got completely different genes, right? So the different parts of the genome are coding for completely different things, yet they share these features. Does anybody have an idea as to why this works?
You have covered this. You covered it in the week that we did, the week before last, at the end, when we solved the problem of what you had, and we said it was not different amino acids, but what? Yeah, so it's codon preferences. Yeah.
Yeah, I think it's actually quite a bit more fundamental than that.
Well, that's part of it, but yeah.
Yeah, the way to view it is like a microbial cell is almost like an ecosystem of genes, okay? And you can be sort of well adapted to that ecosystem. And one way is, one thing that characterizes that is the frequencies of different tRNAs, and hence the frequency of different codons, and hence that then turns into a particular signature of k-mers. And it's probably why tetramers work quite well, because think about codons, they're trimers, so it's probably why tetramers are the best for this as well. But there are other reasons as well. I think another one is probably restriction sites. I believe different microbes have different restriction enzymes for protecting themselves from phages. You're going to avoid those in your own genome probably. So I think that's also a part of it. Can anybody give me an example of a gene that would probably be deviant from the expected tetramer frequencies for a particular genome? So which can you give me an example of a gene where we think this might not work?
You mean a particular gene or a class of genes?
Yeah, just a class of genes. Yeah, a class of genes. One that we've talked about a lot this morning. Well, it's the 16S.
The 16S.
Or other structural ribosomes. Because it's not codons, is it? So it won't fit with the expected pattern of codon usage because it's not codons.
So in a GC-rich organism, so I haven't thought about, are the 16S GC-rich as well as all the coding sequences? Or is the GC richness only the result of the coding genes?
I'm not sure, okay, but whatever genome you have, if you basically look at the GC content, there will be deviations. And when you get to the 16S, it will look completely atypical to the rest of the genome. But it's also not just true of 16S genes, but actually, unfortunately, and this causes binning problems, it will be true for any gene that is accessory to the genome, okay? So, as I said, if you sort of view the genome as an ecosystem, the core genes that are shared across all strains have been co-evolving, and they will better reflect the conditions of the cell. But a gene that's come in relatively recently will have an atypical sequence composition to the rest of the genome. And then over time, it starts to become more similar as it sort of adapts to its local environment. So you can actually, it's really interesting when you've got a genome to look at these changes in composition along it. All right, but in general, the point is we can use this as a signal that is useful for clustering contigs. But as I said, because of the imprecision of sequence composition, another signal that we can use is differential coverage. So the principle here is quite straightforward. So imagine we have two samples from a metagenome. We actually assemble them together to form what we call a co-assembly. Okay, so we've assembled all the reads from sample 1 and we've assembled all the reads from sample 2 into a single co-assembly. Okay, then we map the reads back independently. So we map the sample 1 reads onto the co-assembled contigs, and we map the sample 2 reads, and then we compute the coverage depth of the sample 2 reads on each contig and the sample 1 reads on each contig. And this is the, I think you must have covered this, right? This is the equation for computing the average coverage depth on a bit of sequence. We take the number of reads that map onto it, multiply it by the read length, divide by the length of the sequence. Okay. But the key principle here is that if you have contigs deriving from the same genome, and that genome is present in multiple samples, but with different abundances, then the coverage depth of all the contigs from that genome will fluctuate, but they will be correlated in their coverage. And they will be have a different coverage profile to contigs from a different genome. And this principle was nicely illustrated in this Albertson et al. Paper. So they took a single sample, but they did two different extraction protocols. And every extraction protocol has a different set of biases. And they did a co-assembly, computed coverage depths for one extraction protocol on the x-axis and for the other extraction protocol on the y-axis. And you can see you get these distinct clusters that also correlate with taxonomic classification. And that's, as I said, because all the contigs from the same genome have a similar coverage profile. And that's basically the principle to automatic binning. We combine these composition signal and the coverage signal with a clustering algorithm. And clustering itself is basically a very simple process. Simplest forms of clustering algorithms just depend on some distance metric. So I talked about this when we were doing hierarchical clustering of 16S sequences, right? You just need to define a distance metric, and then you can perform hierarchical clustering with contigs. And that's actually what's done in the AMPLIO program. The problem with hierarchical clustering is you always need to decide on a cutoff. So probably for binning, we prefer what's called a partitional clustering type algorithm. This was actually used in the first version of MetaBat. I think MetaBat 2 has moved on to a graph-based clustering algorithm. But I'll just explain very quickly how k-means works. So you've got a set of data points. We're showing these in 2 dimensions here, but it will work in any number of dimensions. And we initially choose k data points as our means of our clusters. You then reassign every data point to its closest mean based on your distance metric, and then you recompute those means as the average over all the data points assigned to that cluster. And then you keep repeating that process. And the amazing thing is, k-means is guaranteed to converge onto a fixed set of clusters. There's actually a nice mathematical proof of that. You still then have the issue of how to define the number of clusters in your data set. And so for that reason, I prefer model-based clustering, and that's how our binning algorithm works. So this is called Concoct. And I'll talk very briefly about how Concoct works. But essentially, the data pre-processing is as I described. You perform co-assembly across all samples. We actually fragment contigs that are greater than 10 kilobase pairs into 10 kilobase pair pieces. We map reads back from each sample individually. We get a coverage profile. We've also got a Kamer frequency profile. So you have zeros in those vectors. we add on pseudo counts, which means we can then, we add on pseudo counts, we normalize coverage and k-mer frequencies, and then we log transform them, and that enables us to perform a PCA. But we don't perform a PCA in order to visualize the data down 2 dimensions. We use it to reduce the dimensionality of the data and also to reduce correlations. So we keep enough dimensions to maintain 90% of the variation in the data. So I'll talk about PCA again a little bit more on Friday, but it's basically a linear method for transforming data that tries to preserve as much of the variation in the data as possible. So each component of the PCA is orthogonal, and it's basically presented in decreasing levels of variance explained. So you can keep enough dimensions, as I said, to explain 90% of the variation. And then we use a model-based approach for clustering. So we actually use something called a mixture model. So this is a statistical distribution which describes a more complex distribution as the sum of simpler components. So each component in our distribution is a Gaussian. And what that corresponds to in terms of clustering is the assumption that you have you're describing the cluster as an ellipse. But we use a completely general Gaussian, which means our ellipses can be of any size and any orientation. One of the drawbacks of the k-means algorithm is that you're actually assuming that each cluster has the same size. And for various reasons, the amount of noise in a metagenome bin will vary from one organism to another. Things like GC content, also the overall level of the overall coverage. And then, so to deal with this question of how many clusters are present, we use something called automatic relevance determination. So we start off with more clusters than are needed, and the ones that aren't needed will shrink out of the data set. Now, Concoct was, I think we devised that in 2015. We published that paper, and it was one of the first automatic binning algorithms. It was probably the first one that actually worked well, and so it created quite a lot of interest. But it's actually really easy to devise a contig binning algorithm because it is just clustering, and any computer scientist can come up with an alternative clustering algorithm. So these days there's hundreds of them. I'll just give you a brief overview. One that is very useful is MetaBat 2. It's a lot faster than Concoct and gives comparable results, as does MaxBin 2. I'm not so convinced by Coca-Cola, though it has some nice principles. VAMD is super interesting. So it's based on a pre-processing step which isn't PCA as in Concoct, but uses something.
You're getting a bit stuck in the
variational
details here,
autoencoder.
Chris. We need to move on, I think, because it's already 3:15.
All right, but honestly, I mean, what is the point in trying to teach people if we don't teach them the details? So, I mean, what are we teaching them?
We're teaching the
Okay,
basic
I think
principles.
they need to know different
Okay.
clustering algorithms because
They're
they
not-
might want to choose different choices when they do their own analysis. But I will speed up, yeah.
Sorry, can I just ask, I missed, what did you say that Concoct assumes when you were forming the bins?
Say that again, I didn't
She
catch
wants
that.
to know, what does Concoct assume when it's forming bins?
Okay, so it's basically trying to find clusters that are nicely represented by ellipses. Okay, in terms of how it does that, it is a form of EM algorithm. So you take each data point and you say, what is the probability that it derives from this cluster or that cluster, given an error model that we're learning. So it's sort of like, you can see its connection to k-means, where you reassign clusters and derive the means. But as I said, it enables you to have a flexible shape for each cluster. The other advantage of it, and it's one of the reasons it's still used in certain contexts, as I said, despite being quite old, is that it has no prior assumptions. So MetaMap2 is sort of makes an assumption about how divergent different bacterial genomes should be. A lot of the other algorithms use single copy core genes as seeds, for instance. But Concoct makes no assumptions whatsoever. So a Concoct cluster can actually do a decent job of clustering out viruses or eukaryotic genomes as well. So it sort of means that it has some meaningfulness outside of just prokaryotic genome bins. We still like it in a couple of contexts. Oh yeah, that's what I would have said there. Okay, all right. There's also a couple of newer algorithms that are based on also using information from the SM graph. Example would be GraphBin or MetaCoag. Once you've formed your bins, then you use these single copy core genes to evaluate them. So you look through each bin. You say, is it complete in terms of single copy core genes? Does it seem contaminated? And then with a certain sort of cutoff on completeness and contamination, you decide whether you're going to keep it as a MAG. All right, I won't talk about when you should do co-assemblies of single samples. And I won't, so I'll very briefly show you the results from binning on the CICRA data set. So this was quite a low data set, but we still got a reasonable number of MAGs, some 268. And we're able to do so this is, I think, Mark will talk about this a bit more as well. One of the nice things about pulling out MAGs is you can now do a high resolution phylogeny. We use those core genes concatenated together to build a phylogeny of all the MAGs that we've obtained. But we can also use them to profile the community in just the same way as we profile with taxa or function. So here I'm showing an NMDS, but based on Concoct clusters. And red is individuals with Crohn's disease, green is healthy individuals, and just as we were seeing with the 16S, you can distinguish between the two groups. But yeah, I'll skip through this in the interest of time. Okay. So yeah, so hopefully that was a useful overview of how you do metagenomics binning, and yeah, now you can go on and do your tutorials. You can have all the PowerPoints.
I've recorded the talk as
well
well.
Later, if you're actually interested in the details, which apparently you're probably not.
No, some of them are, but some of them are not. That's the point. It's a general audience, Chris.
Well, then I sort of feel that people that aren't should just sit, can sit there and
Okay.
Anyway, fine.
Okay.
So look, I think that's me done for the day, isn't it? You can do the tutorials now.
Yeah. Okay.
Right, and I'll see you again at 10 o'clock tomorrow.
Okay. Then.
Thanks a lot. Bye-bye.
Bye.